So thank you so much. I'd like to say I'm very grateful for Doug Hilton, who was on my selection panel for Dean, because not only did he help me become Dean, but the current Vice-Chancellor asked me in my interview, of course I was going to give up research because I couldn't be a Dean and do research, and Doug Hilton leant across the table and said, Ed, surely not, surely you want your Deans to be research active. And Ed said, yeah, of course. So I still have an active research lab, and I'm very grateful, as always, to Doug. So I'm going to talk to you about some published work we've done on tumour suppressors in breast cancer, and a new tumour suppressor we've identified in breast cancer, which is unpublished work. So the pathway I've worked on since my postdoc, after doing my PhD with Hatem, is a pathway mediated by an enzyme known as the PI3 kinase, which is mutated in many human cancers. And it makes a signal called PI345P3, which is a lipid signal that is only there really in stimulated cells in cancer cells. And so although phosphoinositide metabolism has been known for 50 years, this signal was overlooked until Lou Cantley discovered it in the late 80s. And many people didn't believe it existed. And in fact, I remember Phil Maduris in my postdoc lab said to me, Lou Cantley's got it wrong again. But Lou Cantley actually examined cancer cells where this signalling pathway is hyperactivated. The enzyme that makes this signal, the PI3 kinase, has a P110 catalytic subunit, and the gene that encodes this subunit, PIK3A, is mutated in up to 50% of all human cancers, and it's mutated in 20% of breast cancers. The lipid signal, PIP3, is only a transient signal in normal cells. It can be degraded by two potential metabolic pathways. One is by the tumour suppressor P10 that takes off a 3-position phosphate, and loss of P10 causes both inherited cancer predisposition syndromes and is an acquired sporadic mutation in up to 10% of all human breast cancers, more so in prostate cancer and other cancers. Or it can be metabolised and degraded rapidly by another group of enzymes known as the 5-phosphatases, which I worked on for the last 20 years, to make a new signalling molecule, PI344P2. Now, the downstream effectors that mediate these signals that lead to hyperproliferation, prolonged cell survival, and alter cell motility and metastasis are mediated by a variety of effectors. The one I've listed here is the serine-threonine kinase AKT, that bind to PI345P3 and 34P2, leading to their allosteric activation. So in cancer, you have high levels of 345P3 and high levels of 3,4. You have hyperphosphorylation and activation of AKT because the levels of these lipids are high, leading to a canonical cascade that promotes cell growth and proliferation. So you can get hyperactivation of AKT because you've got oncogenic mutations in the P110 catalytic subunit, because you've lost P10. But there's very little evidence, apart from the unpublished story I'm going to tell you today, that the 5-phosphatases, which I've worked on for 20 years and hoped would be tumour suppressor genes, would be tumour suppressor genes. So they're kind of enigmatic. If you actually lose them, as I will tell you, you get a whole variety of developmental abnormalities, but not cancer, except for the one I'm going to tell you today. This signal, which activates AKT, is degraded by an enzyme family known as the 4-phosphatases, which remove the 4-position phosphate to make PI3P. And we and others have recently published that they are tumour suppressors. And I'm going to tell you the story of why they're tumour suppressors and which cancers they're affected. So why is this important? There are a lot of studies in which phase 1 and 2 clinical studies looking at PI3 kinase inhibitors for cancer, and they're looking at cancers where there's oncogenic mutations in the P110 catalytic subunit or loss of P10. But we would argue that loss of the 4-phosphatase gives you exactly the same phenotype and should be screened for in many human cancers. This is the complex downstream signalling cascade, which I'm not going to talk on, that's activated by the PI3 kinase signalling pathway. The net effect is to prolong cell survival and promote cell proliferation. The 4-phosphatases work here to degrade this signal, PI344P2, which the pH domain of AKT binds to, leading to its allosteric activation by phosphorylation by other kinases. So we would predict that if you lose the 4-phosphatases, this signal will go up and AKT will be hyperactivated leading to prolonged cell survival and cell proliferation. And if you lose the 4 phosphatases, of which there's 2 forms, they should both be tumour suppressor genes. The 2 enzymes look similarly. They've got an N-terminal C2 domain. There's a type 1 and a type 2. They're named A and B by the Human Genome Project. This is their gene name, and their 4 phosphatases as their protein name. Now, 4-phosphatase 1, we and Sasaki made a knockout, and it was published in Nature 2 years ago. If you knock out IMPP4A, you get a brain phenotype with a Parkinsonian type phenotype associated with clonic tonic seizures. There is no evidence that IMPP4A is a tumour suppressor. However, we and others, and I'll show you this study, suggest that IMPP4B is a tumour suppressor that's probably just as important as P10 and has only been recognised in the last 2 years. So we started studying IMPP4B by making a monoclonal antibody to the protein to look to see where it was expressed and what happened to it in cancer. And we're interested in breast cancer. So we, by a lot of effort, we made recombinant protein and made an absolutely fabulous monoclonal antibody. And the first thing we did was to look in normal breast. These studies were all done in collaboration with Christina at the Alfred Hospital here. And we noticed that in the normal breast that IMPP4B was expressed in the breast ducts, but not in every cell, in a pattern that was highly reminiscent of the estrogen receptor, which is not expressed in every cell in a normal breast. We then went and co-localised IMPP4B in the normal breast and showed that it co-localised with the ER receptor, and so estrogen-positive cells co-express IMPP4B, which we predicted would regulate cell proliferation by controlling AKT signalling. When we scored the normal breast tissue for cells that were proliferative by co-staining with Ki67, we showed that, or Claire Fidaly in my lab showed that IMPP4B positive cells were estrogen positive and were non-proliferative. Whereas if you have lost IMPP4B, they're very proliferative. So this is highly proliferative normal cells have lost IMPP4B, and the non-proliferative ER-positive cells have got IMPP4B. We then went and did classic studies in which we took a cell line that was ER-positive. We knocked down IMPP4B using sRNAi. We did signalling, and we showed AKT was hyperphosphorylated. We showed that these cells were more proliferative and formed colonies in soft agar. And we showed that when we injected those cell lines into mice, the mice formed bigger breast tumours. Consistent with IMPP4B suppressing PI3 kinase AKT signalling, when you lose it, you get hyperproliferation. So that's all very nice, but mice and men aren't the same, as Katrina keeps telling me. So we then went and did an extensive study by studying 400 individuals with breast cancer. We used 2 cohorts. One was a cohort in collaboration, 293 cases with Grant Sutherland in Sydney at St Vincent's, and 160 plus cases here at the Alfred Hospital. And we showed that there was a positive correlation between IMPP4B being expressed in ER-positive breast cancers and it was lost in ER-negative breast cancers. It was present in PR-positive but lost in PR-negative. It had no association with HER2/neu and no association with EGF receptor. Furthermore, we showed that if you lose IMPP4B, you're likely to have a much more aggressive tumour. It shows a correlation with ER and PR. The tumours are more likely to be proliferative. There was no association with PIK3 mutations, but there was an association between the 2 phosphatases. You are more likely to have lost the 4 phosphatase if you've also lost P10. And when you lose it, you get hyperproliferation. And that breast tumours not only should be screened for loss of P10 or for activation of the PI3 kinase, you should also look to see if they've lost IMPP4B, because these cancers will be targets for PI3 kinase inhibition. Subsequent to that study that was published 2 years ago, a whole series of now studies have come out showing that IMPP4B is a putative tumour suppressor in a number of human cancers, including prostate cancer. It's lost in 47% of prostate cancers, and in unpublished studies, again, we've done with Grant Sutherland, which is very sad. Rob Sutherland, sorry, it's very sad that he's no longer with us. We've shown that IMPP4B is a very strong predictor for survival in prostate cancer. With Andrew Way, we've shown that IMPP4B is differentially expressed in acute leukemia and also between ALL subtypes. That's published, but the studies we're doing with Andrew Way shows it's differently expressed in AML. It's associated with loss of heterozygosity in melanoma. Cantley has shown it's associated with loss of heterozygosity in ovarian and breast cancer, correlated with decreased patient survival. And now many studies have started to show an association between IMPP4B and breast cancer. So we think this is an important new, relatively overlooked tumour suppressor gene that regulates PI3 kinase signalling. And it works here in the pathway. So in the second part of my talk, I'm going to talk about unpublished work we've done looking at some of the 5 phosphatases and why aren't they tumour suppressors. You could argue that although they break down a lipid-promoting signal in PI345P3, they make another signal that promotes cell growth, PI34P2, which is then degraded by the 4 phosphatases. And to date, none of the 10 5-phosphatases have been shown to be tumour suppressors. I purified this enzyme, which was the first one to be purified in my postdoc. I'd spent 3 years purifying coagulation factors and Hatem kept telling me to go back to the cold room. I got to Phil Maduris' lab in the late 80s hoping to learn how to clone. Hatem promised me that was what happened. Phil Maduris took one look at me and said, "Oh, you know how to purify. You can go to the cold room and purify 5-phosphatase." So I came back an expert in purification. Having purified this particular family member, IMPP4B, which we subsequently cloned. When you knock it out, it's got a defective spermatogenesis, nothing else. And then a whole series of other people, because we cloned the next one, discovered a family of 10 enzymes with a homology in their 5-phosphatase catalytic domain. The top 8 have been knocked out, and they really all have phenotypes. They have quite significant neurological phenotypes, SNP1 and SNP2. SHIP1, if you knock it out, gets a myeloproliferative disorder, although there's argument about what the nature of that myeloproliferative disorder is. SHIP2 gives you every woman's dream. You can eat what you like and you stay skinny. I wish I worked on SHIP2. SKIP gives an embryonically lethal phenotype for unknown reason. IMPP5E gives you ciliopathy and embryonic phenotype. Now, the one I'm going to talk about, the 5-phosphatase, is PIP, which the knockout has not been reported, and which very little is known, and this is all unpublished work. So this 5-phosphatase has really been overlooked. We cloned it with others, and we showed that it localises to the plasma membrane. It opposes PI3 kinase signalling by hydrolysing PIP3 to 4,5P2. It suppresses AKT activation in response to EGF, and that's pretty much all that's known. However, we noted that there'd been a lot of gene array studies that had picked up PIP as being a gene that showed differential expression in breast cancer. And some large-scale screening studies published in Nature Medicine in 2002 when they screened 5,000 genes for genes that showed differential expression which related to breast cancer prognosis. PIP was one of the top genes that had been identified to show differential expression. PIP stands for proline rich inositol polyphosphate 5-phosphatase. The gene is located on a chromosomal region that's frequently associated with loss of heterozygosity in breast cancer. And in a more recent publication, another gene array study suggested that PIP was a gene that showed one of 10 highest ranked genes for predicting human breast cancer patient outcome. And we published that in cell lines, PIP opposes PI3 kinase mutations in oncogenic induced transformation by regulating AKT activation. So with Katrina McLean, we embarked on studying the expression of PIP in human breast cancer samples, and we screened 176 breast cancers by subtypes and showed that PIP is expressed in ER and PR positive breast cancers. Its expression shows significant loss in poor prognosis ER, PR positive breast cancers and in PR positive breast cancers, and it seems to be most closely associated with estrogen receptor expression. It shows significant loss of expression in triple negative, poor prognosis breast cancers. and in high-grade breast cancers, suggesting the gene array studies might be right. You might need it in good prognosis breast cancer, and when you lose it, it may contribute to a poor outcome. So we did sRNA knockdown in breast cancer cell lines and got quite good knockdown. We showed that when you knocked it down, you got hyperactivation of AKT, as shown by serine 3 and phosphorylation in the cell lines. The knockdown cells not only showed many more colonies, but the colonies were huge, suggesting that when you knock it out, it does, at least in cell culture studies, promote cell proliferation. And we also showed that if you took these knockdown cell lines, and this is all unpublished studies by Lauren Binge, a PhD student, and Lisa Ooms, a postdoc, you got much bigger tumours. Consistent with, if you lose PIP in breast cancer, the tumours will get bigger, AKT will be active, consistent with the gene array studies. So that was all looking good. We decided not to publish it and to hold out for the knockout mouse. What a foolish thing to do. So we knocked out PIP. Knockout's never been reported. And we waited 2 years for these knockout mice to get breast cancer, and they never did. So we've got all this data suggesting it's a tumour suppressor. We knock it out in mice, no tumours. Yet again, proving many people's studies that mice and women are not the same. So we then decided, we read all the literature on breast cancer and decided for many tumour suppressors you need 2 hits. You need to knock out another tumour suppressor or get an oncogene. So we crossed our mice with a very well-known mouse model of breast cancer, which is the polyoma middle T antigen mice. This activates PI3 kinase. It's under a mouse mammary-specific promoter. And these mice, with 100%, get breast cancer at about 150 days. And we predicted that if we knocked out PIP, they'd get breast tumours much sooner. And we thought they would show accelerated metastasis to the lungs. All these mice get metastasis. And it looked good. So if you cross the PIP knockout mice with these breast cancer prone mice, you can see at day 40 there's much more hyperplasia in the PIP knockout mice on PYMT background. This is accelerated even further at 70 days. They show evidence of hyperproliferation, as shown by Ki67. They show evidence of AKT hyperactivation, but they all get tumours at the same time, and there's no difference in the size of the tumours. So we then thought, well, it obviously is accelerating tumorigenesis, but this is a very aggressive breast cancer model. So what happens to metastasis? Do these mice get more metastasis? Is this promoting metastasis consistent with its role? If you lose this gene, you've got a poorer outcome, as shown by the human studies. So I said to Lisa, go look at the lungs. All these mice get lung cancer. And lo and behold, on a PIP knockout background, we cured the metastasis. We didn't promote the metastasis, we cured the metastasis. And we had a massive decrease in metastasis. So we did all this blindly. Katrina looked at it and she said, yes, they've got a significant decrease in metastasis. So we went back and did what we probably should have done at the beginning, was to read the literature. So it's interesting, PI3 kinase signalling, although it promotes cell proliferation, its downstream effector AKT promotes proliferation and survival, but actually inhibits, at least one isoform inhibits cancer metastasis. So AKT1, an isoform which is most strongly expressed in breast, actually inhibits cancer metastasis by inhibiting cell migration. So we went back and we looked at the migration of our PIP knockdown, and we have done this in the cancer cells in the mice as well, and we showed that if you lose PIP, the cells, instead of migrating in a straight fashion, sort of wander around and can't close a wound, and we've shown in the mice the migration of the cancer cells is impaired. So we would propose that IMPP4B is a tumour suppressor. PIP looks like a tumour suppressor in that it promotes cell proliferation if you lose it, but it appears to be playing a complex role in metastasis by inhibiting metastasis. So all this work was done by Claire Fideli, a PhD student, Lauren Binge, a PhD student, a postdoc in the lab, Lisa Ooms, in collaboration with some of the samples came from Graham Giles's group at the Cancer Epidemiology group, and by Katrina McLean and Rob Sutherland. This work was all funded by the NHMRC. Thank you.